Alrighty, we're going to be talking about DNA synthesis now. And in order to start, we have to take a look and just review quickly DNA. So if you remember, DNA is a double helix, it's a double-stranded helix. Okay, so what this means is it's got one strand and the helix around each other, and the base pairs are between them. Okay, so if we stretch that out and we unravel it out, what we're going to get is we will get a sugar backbone. Remember, it's a sugar phosphate backbone on one side, and we're going to get a sugar phosphate backbone on the other side. And in between them, and in between these sugar backbones, we're going to get the nitrogen bases. So we're going to say A, C, G, T, A, G, G. Okay. So those are the nitrogen bases on one strand. And this is just an example. This isn't 100% set out. Although the probability of this sequence being in your DNA is highly likely. Okay, so that means the other strand is going to be complementary to it. They're complementary strands. That means they're going to bind. So the A binds to T, remember, C binds to G, G binds to C, T binds to A binds to T, G will bind to C, and G will bind to C. And then these are attached to another sugar backbone. Okay, so there's bound to the sugar backbone right here. And remember, the things that are holding these together are these weak hydrogen bonds. These are hydrogen bonds that are holding these together. So that means they're easy to break apart. You can easily break these bonds apart and unravel the DNA if you need to, which is actually the first step of DNA synthesis. So now, why do we need to synthesize DNA? Well, anytime you need your cells to replicate, they have to totally make copies of themselves, and the most important thing to make a copy of is the DNA. So the first step of this is we've got to unwind and we've got to separate. Unwind and separate the strands. And this is done by an enzyme that is called helicase. Okay, and what that does is it sets up what's called a replication fork, and it makes a replication fork. Okay, so if we take a look at that, then basically I want to keep the same things. So we've got their DNA here, and then it forks up. Okay, so we got A, C, G, T, A, G, G on that side, and then T, G, C, A, T, C, C on this side. Okay. Now, if you've done this before in biology, you know that there's this thing called a leading strand and a lagging strand. And I'm really not, I really don't think it's that important for this class to do it, to know that. Basically, what we're going to do is we're going to go to the hallmarks, and just the big picture stuff. So then when we've got a replication fork, what we have to do is we have to copy the DNA. Okay, and there are several enzymes that do this. Several enzymes that do this, and the first one is called primase. Okay, and so what primase does, it's an enzyme, ASE. Remember, ASE means enzymes. So what primase does is it sets out and it builds an RNA primer. Okay, so remember, in RNA, there's no T, so there's U. So U, G, C. And let's just say this is our primer. Okay, so this is the primer. Oh my gosh. Can't write today. Let's just erase them both. This is the primer. Okay, sets up that primer. And then when that primer is laid down, another enzyme, so primase builds the primer, builds an RNA primer. Okay, So when you put down a primer, that's basically the signal. This is where you start. This is where it starts. Okay, and so then we have another enzyme called DNA polymerase, and this builds the new strand. Okay, the new strand. And I'm going to put the new strand in a different color. I'll do it in green. And so this DNA polymerase is going to come and it's going to add new DNA. Okay, it's going to add a new DNA strand here. That will bind through hydrogen bonds to the old strand. Okay, and it's going to do this along the entire length of the DNA. It's going to start at the primer and it's going to go. And so if you want to, you can think of DNA polymerase as an enzyme box that is going to go and is going to synthesize the strand. Okay, the other side is a little bit more complex, but it basically follows the same rules. Okay, DNA polymerase can only go in one direction. And your DNA is actually it's a two-way street. It's got one going one way and the other one coming another way. So with DNA polymerase on one side, called the leading strand, it can just go straight. Once it attaches to a primer, it can just keep going. It can keep going. It can keep going. However, on the other strand, it's called the lagging strand. DNA polymerase can only do it in small chunks called Okazaki fragments, and it doesn't matter. Who cares? Okay, but the same principle applies. So what you're going to do is you're going to get a primer on the other side. It's going to be ACG. Here's a primer. This is the primer here. Okay, and then DNA polymerase in a smaller chunk is going to lay down the other strand. Okay, we're going to get their other strand laid down. Oops, to the wrong side. It's going to get laid down in the other direction, like this. And then what's eventually going to happen is I'm going to get two new strands. And what's going to happen? And so finally, 3. Remove. Sorry. Before we do this, finally, so I'm going to remove the primers and finish the strand. And the enzyme that does that is DNA ligase. Okay, DNA ligase is going to remove primers and finish the strand, and it's just going to seal everything off. And so what we're going to get in doing this is we're going to get two new strands. Okay, so the end result are two new strands that are semi-conservative. Okay, so what that means is, let's say I have a DNA strand here and I have a DNA strand here. So we can just put some amino acids in here. We'll just have a bunch of A's. So to be fast. So A, and the other strand is going to be T. Okay. So if I need to copy this, I'm going to use that. I'm going to unwind it using helicase. I'm going to use DNA polymerase to build a new strand. Okay. And it's going to be a complementary strand. And then I'm going to use DNA ligase to finish it up. And then what I'm going to get, here's what you're going to get. One of the strands will be the old. It'll have one of the old ones. A series of A's, just like in our old strand. Okay. But the new strand, complementary strand, the other strand is going to be new. Okay. See, I built a new strand there. Okay. And then when I copy the other part of it, it's going to be a bunch of A's. And here I've got my old strand that is full of thymines. Oh, sorry. Okay. So you see that. So a semi-conservative. I have conserved this strand and I have conserved this strand. And these two in here, these are the new strands. Okay. I basically replicated DNA. I have replicated the entirety of the DNA molecule. But with each copy, I get one strand of the old. And that's what makes it semi-conservative. It is a semi-conservative process. Okay, so that's DNA replication in a nutshell. Okay, I think the only thing that you have to remember about where it occurs, where does it occur? Okay, and it's only in the nucleus. Okay, doesn't occur anywhere but the nucleus. Okay, DNA can't move out of the nucleus. Okay, if the nucleus breaks down, either the cell is replicating or the cell's dying. Okay, so those are the two things. And with that, that's all I think you really need to know about DNA replication. And we'll do things in lab about DNA replication just to make sure it's solidified in your head. Okay, so next we're going to do protein synthesis.